So I was expecting to know more people here today, but I only know one person. So welcome, guys. Dr. Patrick Kearns. You can't do that in here. You gotta go in the hall.
Okay.
You're in the lab. Sorry. Busted already. So I'm Dr. Patrick Kearns.
I had class with them both last semester. We had math, the data statistics analysis.
Well, it's easier this semester because I actually know people. I know at least like 50 students. Which is good. Last semester, I knew 0 students,
Yeah.
which was it's fine. But it's always nice to know people. Know what to expect.
Yeah.
All right. Welcome back. So, as I was saying, I'm Dr. Patrick Kearns. Obviously, your instructor for the semester, but I'm also the program director of biology here at Fisher. So, your journey, I guess, starts and ends with me at some point now. In terms of me, I just started back in the fall, so still pretty new to Fisher as a community and the policies of Fisher. So, if you ask me for help on some things that I don't quite know how to do it yet, that's just because I'm new, not because I'm inept, just because I'm still relatively new. My background is in microbiology, so I actually got my doctorate. I was going to say across the river, but Northeastern is over there. I got my doctorate in Northeastern. So I'm a microbial ecologist. So I study like microbes in the environment, stuff like that. My expertise is in like data analysis and stuff like that. Just as a this is who I am sort of person. Clearly you're in genetics. I hope you're in genetics. So I thought I already know how to deal with it. I have 2 new folks here. Could you guys just introduce yourself to me? I had to. I just never seen your face, or your mask covered face. That's the problem. I met one of my superiors today, who I've never seen with a mask on, and I didn't recognize because she had a mask on. It's usually the opposite. Usually I don't recognize my students because they have no mask on. I'm used to seeing you guys all the time. So clearly you're in genetics. So we meet 9:30 on Mondays, and we meet for I have the room wrong on here. That's not bad. Which is why I went across the hall. But we're supposed to meet in here. We meet for a lecture for an hour and 15 minutes, and then we stay together for another 45 minutes or so for recitation. I don't understand the point of the recitations, to be perfectly honest with you. It seems like we have enough material to cover that we should have 3 hours of lecture every week for every class. But we'll use recitation for a combination of covering extra topics and giving you guys time to work on problem sets throughout the course. The genetics, as I'm sure you probably know, is inherently problem-solving based class.
Are you going to stop for the 5 minutes, or you're just going to continue
It's going to
through?
be someone like cell biology. If you guys need a break, you can obviously let me know. So this is the syllabus. It's the course Blackboard is all set up. I had nothing to do over winter break. I mean, I did stuff in my house, but I had nothing to do. So I basically split up and set up everything. I made all the problem sets, I made all the slides. So everything is up on Blackboard for you now. You could literally go do the entire course's work beforehand, besides the exams, and right now if you want. So everything is available if you're on Blackboard. It's organized by weekly modules. And every weekly module tells you exactly what you need to do for that week. But at the end of the day, I might have made a mistake on Blackboard. So that being said, always use the syllabus as your end-all, say-all. If you're wondering, oh, when's problem set number 1 due? Check the syllabus. That's the ultimate source of information. So I always tell my students, well, not always, but I try to always tell my students that the syllabus I see as a contract between you and I. This is how I'm going to grade you. This is how I'm going to assess your capabilities as an individual. And this is how we are going to interact with you. So again, it's a contract between us. We're not going to sign it or anything like that. But again, I try to minimize how much I'm going to change this. So in terms of how to contact me, this is my office phone number. My office is literally outside that back door to the left. If you guys ever met with Dr. Zeiger, it's the same office. So, my email is kearns@fisher.edu. In terms of meeting with me, talking with me, just send me an email or come talk to me after class. Be more than happy to meet with you either face time to meet with one another. It's whatever you feel most comfortable with. I'm here Monday, Wednesday, Friday. I won't be here Tuesday, Thursday, just because the college doesn't want full-time faculty to be here more than 3 days a week. It's as best they can. You know, if you have to be here 5 days a week, it's fine, but try to only be here 3 days a week. So if you need me, I'm always available on Zoom. I check my email very, very frequently, except for on like the weekends. I don't expect you guys to really work on the weekends. So I don't try to work on the weekends so I can spend time with my family, something like that. So, but anytime you need me, reach out via email. Happy to help. I also, if you decided like you wanted to call me, my office phone doesn't redirect to my phone, but it gives, if you leave a message, it transcribes the message and sends it to me as an email. So, you know, it gets to the end of the email is the best way to, you know, sort of get at it. So there's, you know, course description, learning objectives. You can read those on your own, but basically it's a genetics course, right? So we're tackling more advanced stuff than you tackled in Bio 120, right, or Science 120, right? Going beyond Mendel. So we'll start with Gregor Mendel in class today, and then we'll expand beyond him, much more encompassing. Talk about how to look at genetic data. So in terms of materials, there's a textbook, Genetics from Genes to Genomes. The newest version is version 7. You don't need to use the most up-to-date version, but they're all the same. Now, you should probably piece together, at this point in your collegiate career, foundational knowledge that you learn in college, like cell biology and genetics and general biology and general chemistry. They don't change, which is why they're foundational. So like what's in the 6th version of this book or the 5th version of this book is exactly the same as what's in the 7th version of this book. They just fix it up slightly. So the textbook is a resource for you. So I definitely recommend you pick up some version of this textbook. Like I think you can pick up the 5th edition for like 20 bucks. And I recommend you pick it up as a extra piece of knowledge for yourself. I won't test you on stuff that is covered in the textbook, but I will test you on stuff that I cover in lecture. So I expect you to know stuff we cover in lectures, and the textbook is there as an extra resource for you to read, if you know, to help you learn stuff that maybe I wasn't so clear about, or you want to know more about.
You're going to post the lectures before class, correct?
Every lecture for the entire course is already up on Blackboard.
Okay.
I told you I went crazy over winter break.
Yeah.
I wrote 900 slides over winter break.
Oh my goodness.
So yeah, everything is already up on Blackboard. Everything. So, but again, the textbook is there for you as an extra resource. So I've been trying to find an open source genetics textbook, like something that's completely free, but I can't find one that's any good. So until I can find a good open source version, we'll stick to an older version of Hartwell et al. For the time being. Yeah, everything's already on board.
Why can I find it? Why can I find it on this?
I did email it to you guys as well. I'll check the Blackboard after effect, just to double check. But everything should be available on Blackboard. Now, in terms of other materials, there's a lab manual available if you want Blackboard already. So it's a lab manual I wrote this semester, or this winter break as well. So we're going to go over, I think, some pretty cool experiments in genetics. So I hope you guys aren't afraid of fruit flies, because we're going to be working with fruit flies for about 5 weeks.
Like the one where we like do the eggs and then they grow. Yeah.
Classic genetic tool.
I saw when Zyger did that one. She had them in the windowsill the whole time.
I'm definitely not going to put mine by the windowsill. They'll die.
Yeah.
But it's all there for you, so you don't need to buy this lab manual or anything. If you want to print it out, I think it's like 60 pages, so you could print it out for not that expensive. And then you probably should have a calculator, not your phone. You might need a calculator for the exams, and obviously you can't have your phone out during. So that's your required material. So try to make it as not as expensive as possible. You guys already spend enough money to come here, so I'm trying to limit that price as much as I can. So how are you going to be graded? Well, there's a number of things. So first is showing up. Participation and attendance is required. Show up, look engaged, try to ask some questions, and you'll be okay with that 20%. Just the way it is. As a general note, I'm not as harsh on participation during COVID, just because I know it sucks. I know it sucks. to come to school with you wearing a mask and the world is crumbling around you and all that terrible stuff. So I recognize enthusiasm is low. So this participation grade is not as enforced as I normally would. So just make sure you show up. Get your 20 points. You know, it's only 5% of your grade, but just make sure you show up. You know, it's an easy way to get 5 percentage points. There'll be 3 exams. They're spaced equally throughout the semester. There'll be 40 points apiece with 40 multiple choice questions. I'm not a big proponent of exams, but if I don't give you an exam, you will not show up to be engaged at all. So exams are there to make sure that you stay engaged with me, with your material, hopefully with your textbook. Discussion boards are 10% of your grade. Those again are already up on Blackboard for you to take a peek at. They're basically having you look at real world genetics problems, not like analyzing genomes or anything like that, but like here's a genetics thing in the real world. Do a little bit of research and write a post about it. That's the gist of that discussion. They're spaced, you know, relatively equally throughout the semester, but they're designed to get you thinking about, okay, how does genetics apply to the real world? Because one of the things I like about genetics, and I approach genetics from a bacterial standpoint, so it's not as exciting as like human genetics, but genetics has so many real world applications and so many exciting real world applications, which is why I like genetics class. So those are there. Nothing crazy. They should take you about 25 minutes a piece. And you definitely want to make sure you write your response out, and then you want to respond to other people's as well.
I'm guessing the discussion boards are due like at the end of the week.
We'll go look at the schedule.
Okay.
Everything's on the schedule. Problem sets. So genetics is an inherently problem-based course. Will comprise 25% of your grade. So the single largest component outside of our friend, the lab. Now these problem sets, there are 10 of them. They are between 5 and 10 questions for you to answer. They're based on the material that we're covering in that general time period. But again, they're problems. They're getting you to think, how do you apply genetics to not so much case studies, but like to data. Right? Because genetics is, again, a problem-solving oriented field. Just, yeah. Now, in terms of what math you will need for genetics, not much. You know, I'm not asking you to do calculus, I'm not asking you to do even algebra. I'm asking you to think about probability, adding and multiplying. That's pretty much it. Nothing. So those of you that are scared, because genetics is inherently a math-based field, don't worry, not a whole lot of math. As much as I would like there to be more math, genetics math is a lot of math. But we're going to stick to the simple math. And then finally is the lab, which I am probably the most excited for because, you know, I have fun pulling it together. So lab is the largest component of your grade, and we'll go over how that breaks down in 1 second. Now, the final thing I want to make mention is there is opportunities for extra credit. So for those of you that have taken a class with me before, you'll recognize this little spiel of what extra credit is. As a way for you to advance your skills as a scientist, as a way for you to feel more comfortable reading scientific literature, you can earn extra credit points in this class, 16 total, by reading 2 scientific papers, not ones you've given me in a previous semester either, and write a less than 2-page synopsis of that primary research part. So that's 16 points. And then as a sort of a genetics thing, there's a single genetics case study about skin pigmentation that's available for you on Blackboard as well. I think it's a pretty cool case study. I found it at the Howard Hughes Medical Institute, and they always do cool stuff. So that's your way to get extra credit. Now, there's a maximum of 24 points between the 2 synopses and the case study. So 24 points is not a lot. Right? It's like 7%, 6% of your grade. So the extra credit's designed to push you from a B+ to an A. It's not designed to save your grade from a D to an A, right? The extra credit is there for you as an individual to bump your grade up to the next level. But again, they're designed to get you thinking about genetics in terms of the case study and get you thinking about how to read scientific articles. That's a really important. Now, in terms of the lab, there's a number of things for the lab. So you have 2, relatively sizable lab reports. One's about Drosophila. So one of our main study organisms is going to be Drosophila melanogaster, the model organism for studying genetics. So we have about a 5-week experiment with Drosophila. It's going to start not next week, I think the week after. Yeah, the week after, I think. where you'll play with these flies, you'll mate them, you'll get some genetic crosses, get some data, and analyze what the expected results are based upon Mendelian genetics. So I think it's going to be fun, but I like flies. So the other lab report is going to be the genetics of waterborne pathogens, which, as I mentioned, I'm a microbiologist, so this one's pretty cool. So this one, you're going to be collecting some water samples or some food samples. You're going to be isolating potentially pathogenic bacteria from them. It's E. coli, so it's not like, really bad like cholera. It's just E. coli. And you're going to be analyzing the genetics of those bacteria, which I think is pretty cool. So those are the two big things. Now, there are a couple other smaller things. So you have a worksheet for what's called the PTC lab. So it's a bitter tasting lab. You might have seen it before. You have a PowerPoint presentation. You're not going to present it. You're just going to make a PowerPoint. That's going to be next week's lab, the virtual genetics lab. There is a diagnosis of sickle cell worksheet, which I think is a really cool one. You're gonna get some DNA. You're going to try to diagnose people based upon PCR. But I think it's cool. There's a worksheet for cancer genetics, which is probably less fun, but I think it's a rewarding experience overall. And then finally, we have lab quizzes. So lab quizzes are going to be pretty much every time you come in. They're basically designed to test to make sure you've read the lab manual before you come in. So within the first 5 minutes of coming into the lab, you'll have a short quiz. It'll be like 3 questions based solely on what we cover, what is being covered in the lab manual. So these are quizzes I will print out and pass out within the first 5 minutes of class. If you are not here on time, if you're more than, I think I wrote 5 minutes in the syllabus, if you're more than 7 minutes late, you don't get to take the quiz at all, you forfeit that quiz. So this is designed to get you coming here on time to make sure you come prepared to the lab. So it's 10% of your overall grade, lab quizzes, 40 points, courses are 400 points, it's 10% of your grade. So show up to lab prepared, show up to lab on time. Again, it's everything you need to know for these quizzes is in your lab manual. Nothing crazy out of the box. Read your lab manual, you'll know everything you need to know. Again, there's like 3 questions. But again, you won't know the answers. Well, you might know the answers, but you should only know the answers if you've read the lab manual. So that's how you're getting graded. Now, in terms of attendance for the lab is mandatory, right? You need to come to the lab. We only meet 13 times for lab over the semester. If you don't show up for lab, you forfeit all points associated with that lab, even if it's an excused absence. Can't have you be making up the labs. There's just no way. So if you are absent, you lose your quiz and you lose anything from that day. So if you didn't show up on the day we covered the cancer genetics worksheet, you forfeit the cancer genetics worksheet. If this was a non-COVID time, I could have you come in on another day, but the reality is I can't. So show up. And then the final thing is just late work. So last semester, and in general, I generally adopt a pretty strict late work policy, just because it's a pain in the, just to be blunt with you, it's a pain in the ass to track down students for late work. Just is. Especially when you have 50 plus students every semester. I mean, that's not a lot of students. I have friends that have 900,000 plus students every semester. So I'm lucky in that sense, but late work is a pain in the butt. So I've relaxed my late work policy a bit as a general note. So I will accept late work 1 week after it is due. So say assignment is due tomorrow. So there's nothing due tomorrow, but you turned it in on Thursday morning at 12:50 in the morning. That would be considered late. You would automatically lose 50% of that assignment's worth. Assignment's worth 10 points. You would start out as a maximum grade you get on that assignment as 5 points. Just the way it is. Now, after 7 days, it's considered late, and you receive a 0 for the assignment. So if you feel like you're going to turn something in late, you're better off using the extra 7 days to work on it before you turn it in again. In terms of late, if you have an excused absence, clearly that's okay. You get COVID, you get into a car accident, someone passes away, hopefully not. Hopefully none of that stuff happens because, it's awful. There's clearly opportunities to make things up. Now, I can't have you make up the labs because, again, COVID restrictions, but we'll figure something else in terms of lab work. But please contact me as soon as physically possible if you have an excuse absence. Just, I don't need to know that, any terrible thing happened in your life. I just need to know something's wrong. I need to know so that I can make the arrangements for you, so that I can be aware that, hey, you're not going to turn this in. And you need time because there's an excuse absent. If you come to me at the end of the semester and say, oh, I couldn't turn this in because my cat died, I can't really do anything. So your best bet with me, and with every teacher for that matter of fact, it doesn't matter where you take classes, just be upfront with your problems. Like I said, I don't need to know everything. But if you're going to turn in something late, or if you're going to try to use an excuse absence, I need to know. So just let me know. I'm happy to work with you as best as I physically can within the confines of fairness to the class. So I can't let you turn in everything at the end of the semester because that just simply isn't fair to the majority of people who turn things on. Right? So the bottom line is just let me know if something's wrong. I can help you out in any number of ways. Now, the other thing I just want to make mention of is if you have a documented learning disability or anything through was it the Office of Disabilities? I don't remember. Yeah, so if you have anything through them, just let me know as soon as physically possible and let me know how we can best work together so that you can get stuff done and whatever accommodations I need for you. I'm happy to help you guys as much as I can. That being said, even if you don't have a problem that's through an official channel here at Fisher, like some severe anxiety, and you have depression, pretend you are facing a struggle for homelessness. Those are clearly not things the college handles, right? But those are clearly things that are impact your life here in this class with me. So as best as I can, I will try. I obviously can't help you with your personal things, but if something is wrong that's not documented through the college, just come speak to me, shoot me an email, let me know what's going on. I will help you as best as I can. But I can't help you in this class if I don't. So if anything is bothering you, and it's making it really hard for you to do your work, just reach out to me, let me know. I'm always here to help students as best as I can. So yeah, please just let me know. I don't need to know the specifics because we're not, I'm not your best friend or anything like that. I'm your program director, I'm your instructor. I'm here to help you succeed. And the only way I can help you succeed is if I know stuff's wrong. So reach out to me sooner rather than later. We can work together, get you through this class, minimize the amount of stress I put on you. I don't want to put a lot of stress on you. That's just not fair. You're here to learn, and if I'm stressing the crap out of you, you're not going to learn. So just let me know how I can ease the stress if something is wrong. Now, that being said, if I'm doing something as an instructor, I'm doing something at all that is putting a lot of stress on you as a learner, again, please let me know. I'm happy to calibrate how I teach. If you don't let me know something's wrong, or if you're not asking me questions, I assume all is well, and I'm just going to keep doing it. But I'm more than happy to calibrate myself as the semester goes on to meet the needs of you guys. Yeah.
As a future reference, do you already have someone as a tutor for this class if needed?
I don't know. I always forget that's a thing. Good point. So, yeah, so like I said, just let me know and we can work it. I'm here as a resource. You don't want to see me as a barrier. That's not how you should see a relationship with the professor. I mean, there are some professors who are jerks, and that's the relationship you have with them. But for me, as your genetics teacher, as your program director, you shouldn't see me as a barrier. You should see me as someone that's trying to help you out. Again, I don't know if my material is not catching on to everyone, if people don't ask me questions. So bombard me with questions. If you think it's a stupid question, just bombard me. Send me emails, come to my office, ask me questions. That's the best way you're going. One of the things I hope you guys, which I don't think a lot of students learn as undergraduates, is that asking questions is like the key tenet of being a scientist. And you should never be afraid to ask questions because all the best things in science history were discovered because someone asked questions. Alexander Fleming invented penicillin. Well, not invented, but coined penicillin as a pharmaceutical drug because he saw something weird happened. He asked a question and he figured out that penicillin was causing his weird little problem. And penicillin, as I'm sure you guys know, has saved millions of lives. Antibiotics have saved millions of lives. And it's all because Alexander Fleming went, that's weird. What's going on? Okay. So questions are your. All right. So this is this course schedule. We're going to stick to it as best we physically can. So today, clearly, course introduction, and we're going to cover Mendelian genetics. So this is a rehash from what you learned in Bio 120. And so each time I give you the chapter that you should read. So in this case, chapter 1. Next week is chapter 2 and 3. And I also tell you what problem set is assigned for that week. So I assigned you a problem set today. It's going to be basic Mendelian genetics problems. Based on what we covered today in lecture, it will be due the following week. So it's assigned today. You'll have it due next Monday. It's not a crazy hard assignment, but because of the way genetics works, there's lots of drawing, lots of not so much equations, but like showing simple math and drawing Punnett squares and pedigrees and all that fun stuff. Problem sets are to be turned in as a physical copy. So
we have to print them out and bring them in.
Well, you're most likely going to want to do it by hand. That's just sort of the way I design the problems. They're designed to be done by hand, and turned in that way. Now the problem sets are available on Blackboard already. I meant to pull up my Blackboard, but I forgot. So the problem sets are.
So do we turn them in every Monday?
Yeah, pretty much. So on the Blackboard here, there's problem sets on the left-hand side, and all they're due is physical copy class. So for instance, problem set 1 is already up there for 10 points, and it's just some problems. They're relatively short. So you can print these out if you want. You can work on them, but again, they're due as a hard copy just due to the nature of the way these problems work. They oftentimes have you drawing something and stuff like that, and that's just kind of a hard thing to do on Blackboard. Now, pretend something bad happens and you forgot your copy at home. That happens. Or you forgot it, you left your bag somewhere on campus and you can't get it. And you want to send it to me as a physical file, do not under any circumstance take a picture with your phone and send it to me. That just doesn't work. Just trust me. I've had a long history of telling students you can't take pictures of things on your phone and send it as a thing.
What if it's scanned? Does
You
that
could-
have the app that can scan it properly?
It depends on how good it is. Because a lot of times students, if I have a difficult time reading it, I'm going to tell you to scrap it. So I'd recommend if you can scan it in. So there's a copier on the 3rd floor. You can scan things in as well.
Is it possible to fax it to?
I don't have a fax number, but
Okay.
if you, like I said, I just don't want, because I've dealt with this for years. Taking pictures of assignments and submitting it, it's so hard to read, and it causes me so much grief to figure out grade 1 really bad picture.
I have a printer at home that faxes copy and all that, so I don't know.
I can look into that if you want.
Just
But again, they should be brought into class. So you'll have a little bit of time every week to work on the problem sets in class. Just as a note. I don't expect our lectures to cover the full recitation and lecture portion, but so you have a little bit of time in class to start working on it, if you so choose.
I saw on the syllabus that we don't have lab on this Wednesday. So are we still coming in for a lecture?
No. So this Wednesday won't have lab just because it's just easier not to. I have a lot of ordering to do today, so I wasn't supposed to come in on work, so
yeah.
Couldn't really say, oh, I needed this stuff.
Yeah.
Can I ask you a question in general? I have some classes that say CoLab or OL lab. Does that mean that the lab is online?
Yeah.
I think
And
so.
the CoLab means that the class will also be online?
So it's pretty much if it says collab, it means that it's online and in class. Unless it does not have a room number and a time.
Does it
that
have
it- So then it's all online.
Okay, cool. I just wanted to make sure.
Yeah.
Sorry.
No worries. Good question. Yeah, so we won't have a lab this week, but we will start next week. Virtual genetics lab. And just go through the syllabus and you'll see everything is there to do. So you'll see that every time there's LQ, it's a lab quiz. It tells you that your PowerPoint for VGL is due the following week. So everything is in terms of due dates is already on here. For instance, you'll see exam number 1 is 6 weeks in. Exam 2 is 11 weeks in, and exam 3 is at the end. And all laid out there for you.
And those exams will be in class, right?
Yep. They're 100% class. So, and the lab is already set up for you, so you can see, oh, we're covering, on week 6, we're covering Mendelian Genetics of Drosophila, part B. And then the final thing I just want to show you guys is the lab manual. So, lab quiz. So this is the schedule of the lab. It matches perfectly with the syllabus, but you clearly see week 1, that corresponds to week. That's on page 4, and then PT, this is the same schedule that's on the syllabus. But this just tells you the pages, so it's easier. There is a lab safety thing as well.
Wait, so print it out and sign it?
Yes. Yeah, I was going to go over that next week when we got into lab together, but you don't have to worry about that today. Next week, we are doing the virtual genetics lab, so make sure you bring in a computer. Sorry? Oh, sorry. Make sure you bring in a computer. Chromebooks will work. If you scroll all the way down to the bottom, there's an appendix about how to install virtual genetics lab on a Chromebook. So like I use a Chromebook, I still can use virtual genetics lab.
How do you get Microsoft Word, Excel to work on Chromebook?
So you have to use it online. So Microsoft has online. So when you sign into your Fisher email account, there's like a little DOS. Yep. You can click on that, click Microsoft Word, and you can work online.
And the same thing for Excel too? Okay, cool. I can never get it to
I just prefer to use the Google stuff.
Yeah.
That's just I got in a habit during my last postdoc of using it, and I never stopped. So, but everything you need for the lab will be here, step-by-step guide how this works. So, so I think like I said, there's a lot of stuff. That being said, this, for instance, this lab 2, we're doing PTC. There's a whole bunch of background stuff on PTC, but there is part A, which we're covering in week 3, and then part B, which we're covering in week 4. So just as a note, each lab has typically multiple parts, and it tells you which lab part we're doing that.
Where's the lab manual?
It should be. Should it be required textbook and materials? There's an editable version, not an editable version, not an edible. You can eat it if you want to. Print it out and eat it if you want to. I don't care. So that's there for you. Now, some of you guys might have noticed I'm wearing a little earpiece. I'm recording my voice over the slides and stuff I'm saying, just as an extra resource for you guys. So if I say something in class, you're like, oh, what the hell did that crazy guy say? I'll post them to this class YouTube playlist every week, basically. So you'll have them to go back and take a peek at. And I'm doing that just to streamline things, to make things easier. But that being said, don't rely on these recordings. Come to class, be engaged, ask questions, take notes. Just because, I was hesitant at first to do this because a lot of science shows that if you do this, students don't learn as well because they are so heavily reliant on the YouTube recordings. But I figured COVID is a great sort of backdrop to try it and see how students. But again, don't rely on it. But it is there if you miss a class. It is there if you don't understand something to say or you need a refresher. Including this class, because clearly I'm a weird-looking dude with an earpiece. All right. And then the final thing, in terms of, I've gone over the student accessibility stuff. I'll try, as I mentioned, I'll try to get back to you as fast as I physically can. Like I said, on the weekends, I probably won't be the most responsive. There's all this other stuff that's put in here. Now, the final thing is academic integrity. This is a very important thing for scientists. It's an important thing for everyone, but it's really important for scientists. And I can't stress how important it is. Now, science is predicated on everyone being honest with one another. So when we talk about the stuff that we're going to talk about in class today and for the rest of the class, we're trusting that the scientists before us followed the scientific method, reported their data accurately, and didn't do something unethical. So that's important, right? You can see how bad unethical science is, right? Look at climate change denial. Look at anti-vaccination, right? These are things that are anti-vaccination has killed tens of thousands of people. climate change, if we don't do anything to stop it, is projected to kill hundreds of millions of people. So bad science and unethical scientists make really, really bad. So I hold you guys all to very high standards. Everything you turn in should be in your own words. Now, if I catch you cheating on the exam in class, that's a different story. But in terms of what you turn in, your problem sets, your lab reports, if I catch you cheating, you'll get a 0 on the assignment. You'll get, I don't know, have to send a letter to the vice president of the college. I don't want to do that. If I catch you multiple times, then they sort of escalate from there. So the important thing here is when you're writing something outside of class or you're working on a problem set, close whatever it is you're looking at, write it in your own words. Because you don't want to get caught cheating. You don't want to lose 25% of your grade for no reason, right? And I'm a pretty big stickler for it. So, and if you ever have any questions, if you're writing a lab report and you're like, oh, I'm worried that this is plagiarism, just shoot me an email. Happy to take a peek at it and tell you what I think. But the gist is everything should be in your own words. Citations should be there when necessary. So if you tell me that E. coli is a common waterborne pathogen, blah, 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 that information is in the lab manual, that's okay. That's common knowledge. But if you tell me the E. coli genome is blah, 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 and it does this with these genes, you clearly don't know that. So you clearly need to cite that. And that citation is not a substitution for taking someone else's words and passing them off. That's your. Now, the other big thing that people do with plagiarism is take a sentence, remove a few words, and claim it as their own. That is equally not as okay. So we call mosaic plagiarism. Catch people doing that every semester. Don't do that. So bottom line is everything should be in your own words. If you want to work on a problem set with your classmate, I'd say okay. But when you turn in your problem set, you turn in your lab report, whatever you turned in, should be in your own words. Nothing you're turning in the semester will be group. Will be a group effort. Everything you turn in will be on your own. And that's not because I don't want you working as groups. You will be working as groups in your lab. But I want to see what you all can do. I want to hone your skills as scientists, as writers, as mathematicians, as problem solvers. But again, you have to be honest with me, and that starts with you. Okay. Anybody have any questions before we?
Okay, the question is for like the vaccination thing. I have two questions. Have you taken the COVID vaccine? Yes or no? If not, are you going to take it? And how would like the scientific opinion?
You want my scientific opinion on the COVID vaccine?
Yeah.
So I have not taken it yet. I will take it the second it is offered to me. Now, the vaccine is, I did not notice it's a bit of water. Anyways, I gotta call maintenance again. The vaccine is safe. There's been tens of millions of doses given out. The only people that have had any adverse reactions to the vaccine are people that have severe allergies. Those people that have allergies are already gonna have a reaction to even the safest of vaccines, like the MMR. So there's nothing to really worry about in terms of this vaccine. It is an mRNA-based vaccine, which you guys probably have read by now, is messenger RNA is what the virus has as its genome. Right? We have DNA, the virus has messenger RNA.
Do you think people that have already had COVID should still get the vaccine?
So the vaccine seems to protect against the newer strains. They're not quite sure yet, but it seems to.
What if you had the newest strain?
Did you get tested for it? Like, did they know, like, did they analyze the genome and say, hey, this is the brand new strain? Or
Well, my stepmom had the first strain, and then I had COVID in the beginning of the month.
Well, it might not be the newest strain, right? Not a high prevalence of the new strain in the United States right now. Or I should say the newest strains, because there's one in South Africa too, and there's one came here from England, but it really kicked up. So, in terms of the technology, you should feel confident about it. You should feel confident because it's based upon the scientific method. That scientists, this is developed in 9 months, but you got to remember that there was decades of work on this group of viruses, right? Severe acute respiratory syndrome viruses. We've been doing work on these for decades. So there's a lot of work behind what went into make this vaccine. So don't let anyone tell you, oh, it was made in 9 months, I don't trust it. That's just not true. There's decades of research that went into this. And the reality is, it's not an inactivated virus. It's not a weakened virus, right? It's not an attenuated vaccine, right? This is just a piece of messenger RNA. So it has no capability to make it inside your cells, and it has no capacity to infect you. That's what's really cool about this new technology. It doesn't have any capacity to infect you. So for instance, the MMR vaccine, that's a live attenuated vaccine. It's a weakened version of the measles, mumps, and rubella viruses. That has the capacity to infect you. It has the capacity to mutate and become measles, right? This one has no capacity to physically do that. So they think this is not only the future of vaccines, but it's going to be one of the safest vaccines. So in my professional opinion, and the professional opinion of some of the scientists that are leaps and bounds better than I am, Dr. Anthony Fauci is the world's greatest, one of the world's greatest scientists on this. He says it's safe. And as scientists, you guys should know. Scientific method works. The reason we have the MMR vaccines, the reason we have antibiotics, the reason we have all this cool stuff, because the scientific method works. Scientific method is awesome. Now that being said, there's lots of bad science of it, right?
I know I'm asking a lot of questions, but technically, my job isn't that I qualify for it, but I work with kids. In a certain way, I'm not sure if I should take it because I've never had it, and the kids that I'm with are never really exposed to it. But because I'm qualified, they're most likely pushing me towards getting it, and I'm not sure how to feel about that.
Well, I mean, at the end of the day, I mean, it's a safe vaccine and it provides you future protection, right? And you're of an age group that has, honestly, I'm close to that age group, so I kind of flow there. That's not really of high concern. Now, that being said, there's still been hundreds of thousands of people your age that have died from this virus. So it is really, should you get the measles vaccine if the prevalence is low? Well, yeah, because it just protects you against future things. And you know, you might work with little kids, but it still might be, they still easily be exposed. Kids are stupid as hell. Like I went to the Red Sox game a couple years ago and I saw a kid licking the handrail, picking up hot dogs they dropped on the ground, which I would do too. Hot dogs, Fenway Franks are awesome. But kids are dumb. I mean, I can't make a decision for you, but in my opinion as a microbiologist, seeing what all the best data says, it's safe and good, even if you've already had it.
Should you quarantine after getting the vaccine then? You're
No.
good, right? Even if you have symptoms after getting it?
I mean, the symptoms are going to be vaccine-based symptoms, right? You're not going to develop COVID symptoms, right? You're going to get, you know, localized redness and swelling. You might get pain in your arm. Those are like
a normal vaccine.
one in say 70 chance for every vaccine. So those are common. Like my sister's got it. She works in a medical center up in Rochester, and you know she couldn't move her arm very well for a couple of days. That's just a normal vaccine thing. That's what vaccines do. They elicit an response, which can create swelling and pain and all this. Now there are more serious things, but you generally would see those really quickly after taking the vaccine.
And when you see those reactions sometimes with other vaccines, usually first.
Yeah, you'll know. So if you're going to have a reaction from COVID, you probably had a reaction from another vaccine.
Yeah.
But the vaccine will be 100% protect you from getting it.
So
Or
it's
99%.
So no vaccine is perfect, but what we do know is that it will help slow transmission. If we can reach a high enough threshold for vaccination, Dr. Fauci says 70+%, which would be the herd immunity level over here, we can meaningfully slow the spread of the virus and protect people who even don't want to get the vaccine. Now that's selfish in my opinion, but it should help slow it down. And that's what's important, right? 400,000 people have already died here in this country alone, right? And if we had the vaccine early on, those people would have died, right? We don't see mass casualties from measles anymore like they used to because of the MMR vaccine. So we can prevent meaningful amounts of suffering and medical bills and all the terrible stuff by more and more people getting vaccinated. Even if you already had it, even if you're not in a high-risk group, you know, you get the, you know, one of the reasons you get some of these vaccines is to protect the most vulnerable, right? We get, you know, a lot of people will get, you know, the tuberculosis vaccines to protect the most vulnerable, the elderly and stuff like that. Just because that's the name of the game, right? Again, always listen to what other scientists are saying, and you know,
Is it ruined all this stuff?
no, nothing's ruined. It just
Isn't it all glass in there?
it's tubes and stuff. Yeah, exactly. So you know, always listen to what the people are, you know, scientists are saying. You know, that's the key. Right? If you want to know something about a science issue, ask a scientist. Don't ask your crazy neighbor on Facebook, right? Right? Dr. Fauci says something's good. If the director of the CDC says something's good, the director of the FDA says something's good, they have mountains of scientific evidence behind And you know, I'm not as good as those people, but I know a lot. So, you know, I know enough to trust people who know way more than me. So like, I don't argue with my wife about finances because she's got a master's in business. I know better. But she knows better than to argue with me about vaccinations. All right, does anybody have any questions before we jump in? All right, so title of talk is Mendelian Genetics. Who's Gregor Mendel? Anybody remember? Arguably one of the most famous scientists of all time, up there with Charles Darwin. Who's Gregor Mendel? Why do we have an entire talk entitled Mendelian Genetics?
I don't want to say the wrong answer, but wasn't he the one with he helped with the Punnett square? Was that him?
Well, yeah, you're getting on the right track. Punnett squares, that's Mendel's area.
Yeah.
Gregor Mendel. Why are we wasting so much time talking about Gregor Mendel? We have a whole lab next week on Gregor Mendel. All right, we'll get into Gregor Mendel in a second. But when we think about genetics, it's the science of heredity, how things are passed on from parents to progeny. Why do you have the genes that you have? That's genetics. Right? How we pass things down. Why do you can answer some of these questions? Why do some children look like one parent while others look like their great-great-grandparents? Yes.
I have a question. I know you said like last time I had you that like the whole ancestry.com and all of those things aren't really 100% reliable. How is it possible that my dad and my aunt, me and my aunt almost have identical like the ancestry, but my little sister is like completely the opposite of us, doesn't have like half of what we have. She's like almost 100% Irish.
Well, that's where you have two key principles. One being independent assortment from Gregor Mendel, right? Genes sort independently on chromosomes during meiosis. And you also have recombination, right? Crossing over. Right. So I think I mentioned this last semester to my cell biology class, but one of the amazing things about sexual reproduction and meiosis and crossing over in those processes is they create, every embryo is a 1 in 1 trillion chance of being unique. Being identical to another embryo that was already created. So you and your sister might be clearly very closely related, but again, you are almost certainly unique. And it just all depends on how those alleles got sorted. It all depends on that crossing over events, all that. So you can look at my sister. I should put a picture of my sister and I. Sister and I look nothing alike.
That's weird. It's weird though, because me and my sister look a lot alike, but we have almost completely different like DNA.
So in terms of ancestry, .com and those ones, they rely on what's called single nucleotide polymorphisms, and they use probability distributions to show that this single nucleotide polymorphism, i.e. change from an A to a T or a C to a G, right, that sort of change, is associated from being with this region of the world. Now, those work decently well from Europe. Less so from Ireland. They work decently well from Europe. They fail miserably in native populations here in the United States. They fail miserably in any population from Central and South America. And they fail miserably from Africa and the Pacific Islands. Because they're all based on single changes being correlated with this ancestry. Now, clearly you and your sister look similar, so you should have some similar underlying genetics. But again, there's going to be some variability in your gene. Thanks. I could talk a lot about this. I hate people that argue with me about them. My neighbor argued with me about how her dog is 100% this. Anyways, so other questions you can answer is what causes similarities and differences in appearance and the skipping of generations. So why am I losing my hair, but my dad at the age of 65 has a full luscious head of hair? Jerk. Right? But genetics provides explanations for the underlying genetic code, how it's passed on, how it influences traits, and explains that all the mechanisms that determine the inheritance of trait. right? So genetics are the basis of heredity. A gene, as you guys should know, is a region of DNA that encodes for a protein or potentially a small piece of RNA. Heredity is also the way that genes transmit from parents to offspring. And the genes obviously underlie the formation of every heritable trait. Hair loss, eye color, height, things like that. And even traits you don't see, like your enzymes. And so for the general themes of Mendel's work, who is the father of heredity, who is the father of genetics, he has 4 general themes of his work. So variation as a whole is widespread in nature and provides continuously evolving diversity. Now that should sound familiar to you guys. That should sound exactly as a line out of Charles Darwin, Origin of Species, right? That's one of the key principles of evolution. Now, Mendel and Darwin were contemporaries. They never spoke, and they did not know about each other's work, but they were working on, unbeknownst to one another, different sides of the same exact coin. Darwin was looking at the change over time. Mendel was looking at how it changed. Now, modern day times, if this was still going on, Mendel and Darwin would be contemporaries. They'd be working together. But 1850s is not exactly the greatest time for scientific cooperation. Now, this observable variation is also essential for following genes from one generation to the next. You can't, if you're studying a trait, it helps to see that blonde hair goes from this generation to this generation, right? A lot of genetics is seeing the manifestation of your genes. Right? If the gene doesn't manifest as anything, well, kind of hard to study. Variation itself is inherited by genetic laws, which can explain why like begets like and unlike. That was a terrible way to say that. Basically, things are inherited in a predictable fashion. And Mendel's laws apply to all sexually reproducing organisms, not bacteria. But bacteria obviously don't reproduce sexually. So this is Gregor Mendel. Now, Mendel came about at a really interesting period of time. People were, it was after, it was age of enlightenment, right? You started moving away from this just like suppressive narrative of church being really heavily linked to state. And people were starting to come up with brand new ideas about science, about math, all these amazing things. And that's one of the cool things about when Mendel came up, is he came at just the right time. So you can imagine Mendel's ideas, if they came, a few generations ago, or Darwin's ideas came a few generations ago, right? They might be in big, big trouble. But they came about just at the right time. And Mendel, he worked on the peas. So you guys probably remember that. Gregor Mendel is classically known for his peas. But Mendel was looking at artificial selection. He was thinking about traits that were selected for in artificial capacity. So what do I mean by artificial selection?
Like you did.
Yeah, I physically select for that trait. I want my dog to have this awful nose that it looks like it ran into a wall. I select for that trait. They're like a pug. Feel bad for pugs, right? Purposeful control by mating choice of the parents, right? Domestication, that's artificial selection. Domestication is pretty crazy, right? You guys, I'm sure, have seen, what like wild versions of corn look like, or wild versions of lettuce look like. Domestication is wild. Artificial selection is wild. Now, the interesting thing was at Mendel's time that despite the use of this artificial selection, of selective breeding, it was super patchy. They didn't quite get, right? No one had really thought, at least in a scientific capacity, how selection really worked, how their artificial selection actually came to be. And so there was a couple misleading theories of inheritance at the time of Mendel, which, is kind of interesting. And this was leading people to misinterpret their selective breeding problems. So the first was the homunculus, which is inherited features of offspring are contributed by the male parent via fully formed miniature offspring in the sperm, which sounds so stupid, right? But if you think about this in the 1850s, okay, I can believe that. Right? It's not like of the time where we believed the Earth was flat, but it's a time of we didn't quite know, right? DNA wasn't a thing then, right? So I can kind of understand this. It sounds so stupid now, right? Right? A fully formed person in sperm. But that's what they do, right? And that's why it was so important to have bloodlines, right? blah, blah. And the second was the blended inheritance, which I think is much more common of what people actually think nowadays, that you're just a blended mixture of your parents. Right? So blended inheritance said parental traits become mixed and permanently changed in the offspring. And I think that's much more reasonable than a tiny person developing in sperm. Again, I just, ugh. People, man. But again, this is blended inheritance makes a little bit sense, right? You might not look exactly like a perfect mixture of your mother and father, but you probably have a feature that resembles your mom, right? You probably have at least one feature that resembles your dad, right? So that's a little bit more reasonable, but again, not quite right. And I think your example highlighted perfectly, right? Ancestry is not quite the same between you. So this is where Mendel came in. He was the first scientist to do this, which is why he's called the father of modern genetics. This idea of the father of heredity. He inferred genetic laws about the appearance and the disappearance of traits in his different generations of his pea plants. So he did peas, as I mentioned, and peas are really good. They grow fast. They have readily observable traits. They're easy to self-cross. They're easy to maintain. So they're great model organisms. Now, Mendel published a paper which is called Experiments in Plants Hybrids, published in 1866, and it became the cornerstone of modern genetics. Now, Gregor Mendel was Austrian, so it was published in, I think German actually. I was gonna say Austrian, but that doesn't make sense. I think it was published in German, but you could read this paper if you wanted to find it. It's in English now. So if you really wanted to take a peek at what Mendel was saying, you can find it. But he used the garden pea, so Piscium sativum. You guys have probably eaten it before. Just nothing exciting. I don't know. My sister thinks peas are the best, but I don't know that peas are much. They're good with others. Now, you might remember from biology too that peas have a pretty, plants have angiosperms have a pretty distinct anatomy. We have, the petals and the stem and all that fun stuff. But most angiosperms, or flowering plants, have a stigma, which is going to be, essentially the female apparatus of the plant, which accepts pollen. And that is a little tube that runs down to the ovules, which are the eggs. Right? And then they also have anthers, which are little pollen sacs. So a flower is both a male and a female at the same time. And you can use, what Mendel would do is he would use a little paintbrush. He would grab pollen from the anthers, and he would pollinate another plant by simply tickling the stigma. Right? Essentially mimicking what a bee does. Right? If you think about what a bee or a beetle or a fly or any pollinating insect does, they touch the anthers, right? And they bring that pollen to another flower. And that's all Mendel was doing. He was just mimicking what bees do naturally. Fun fact, bees serve the American economy at about $1 trillion every couple of years. So just free pollination, about $1 trillion. These are awesome. And so once you pollinated things, a seed would form. Right? So when you pollinate an egg, a seed forms from a plant, and that seed could then be germinated into a fully formed plant. So basic plant stuff. Nothing exciting, but that's Mendel's. And he could control this. Right? You can keep insects out as best you can, and you can control your genetics. You can control, I want this plant to mate with this plant, and no other plants. And you can do that. Because once you cross-pollinate, you snip off the anthers, no more pollination. It's cool. So it's a really nice system. Now, Mendel started off with some key things. So he started off with pure breeding lines of peas. Now, what do I mean by pure breeding lines? Should ring a bell. Michael, I don't quite remember, but I remember the name. What's a pure breeding line?
So like purebred?
Yeah, what is a purebred?
So it's like when I don't know, the best way I can say is give an example. Like how you're saying the pug. They would be a purebred type to get a specific look or get a specific outcome. So like the pugs were bred to the point where like now their face is all smushed in because people kept specifically breeding them with other dogs whose faces were so smushed that now the pug is like flat faced.
Getting on the right track.
I wouldn't say like on a genetic level they have like dominant genes.
Okay, so on a genetic level, the dominant genes, you're both heading in the right direction together. What else? What do you mean by pure? You guys are on the right direction. Only dominant genes, traits. How can I put those two ideas together?
So like the dominant trait mixed with dominant trait.
Yeah, so you only have one type of trait, right? I only have weird, stupid, smush-faced pug. Pugs aren't stupid. The breeding of what we did to pugs is really bad. They have all sorts of problems. Right? I only have that type of genetics inside. That's what we mean by pure breeding. Now that can be dominant, or it can be recessive, but it's pure genetic. Right? I only have one allele of a given trait. That's what we mean by pure breeding. And he controlled this pure breeding idea through crossbreeding, or sometimes even self-fertilization, which he called selfing. So I take the anther, the pollen of one plant, and I fertilize itself. So I was going to say plant incest, but that's not quite right. But you're just basically making a baby with yourself. And so he controlled his breeding. He produced large population numbers, and the traits remained consistent throughout time. So he always knew he had a pure breeding line for the traits he was interested in, which is important. Pure. And he also found inheritance of alternate forms of traits. So we find sometimes what we call antagonistic pairs of either or traits. So for example, purple or white, or yellow or green. So pure breeding for green or pure breeding for yellow. And he had some really awesome experiments. And you know, people like to think of Charles Darwin as this guy that went to the Caucasus Islands and studied a bunch of stuff and published a book. But like Gregor Mendel, Charles Darwin was an incredible experimental biologist. So, and I say this, you don't get that if you've ever read Origins of the Species. If you read Darwin's book about earthworms, I know it sounds awesome, but trust me, if you read Darwin's book about earthworms, you see how brilliant of an experimental scientist Darwin was. And Gregor Mendel was like that. He was brilliant. He had so many good controls. He had carefully planned out experiments. He had carefully controlled breeding. Like he was mental about it. Like the dude ate, slept, and lived peas doing his experiments. And he always, importantly, analyzed things mathematically. Now, I know math isn't everyone's favorite topic. And frankly, I don't particularly like math either. But math is essential too. And that's the important thing about it. You can observe all the things in the world, but if you can't test it mathematically, it's not that good. And genetics is inherently a mathematical field. Did you have a question?
I was gonna ask, did Mendel eat his experiments like how Charles Darwin did?
Yes. Yeah, no, he didn't let his piece go.
Yeah.
He ate his piece. I mean, why would you? Now, Mendel had, as I mentioned, antagonistic pairs, either or. So I'm sorry if this is kind of hard in the back to see. I thought I was going to be in a smaller room. The biology one. I didn't look at the room number. Like yellow and green. You know, pea shape, round or wrinkled. Flower color, purple and white. Pod color, green or yellow. Pod shape, round or patched, like teeth almost. Stem length and flower position. So we had all these traits you could look at through time, easily to see. And again, peas are a good model organism. They're produced fast. Traits are readily visible, but easy to energy. Now, he did what we call monohybrid crosses. So you probably remember what a monohybrid cross is. So Mendel crossed pure breeding lines that differed in only one trait, for example, seed color. So he had two pure breeding lines, yellow peas and green peas. So he'd take the sperm of yellow peas from the anthers, and he fertilized the anthers of our green peas here. Again, pure breeding populations. Now, it's important to take note, he had the P generation or the parental generation, where this all started. The important thing to remember, parental generation, that's where your experiment starts first. Now, he would produce what we call the first filial, or the F1 generation. One of the most fun words to say in science is filial. And his monohybrid cross, again, looking at just one trait, yellow versus green, his F1 generation would be all yellow. Okay. So, what did he do after that? Well, he took the sperm and the egg from these yellow peas and crossed them. And he got his second filial, or the F2 generation. And what he got was 6,200 plants with yellow peas and 2,000 plants with green peas, which, if you are a math inclined, is approximately a 3-to-1 ratio. Again, this is just stuff you might not remember it from general biology 1, but again, this should be clicking around in your head as something that's relatively familiar. And so, what you're seeing here is this really simple cross, right? 2 traits, pure breeding lines. He established them through continuous breeding. You get his first filial generation. They're all yellow. He crosses F1 generation, getting an F2, which was a 3-to-1 ratio of yellow to green. Now, what this does is disproves the hypothesis for blending. Now, what would the blending hypothesis look like? Remember, blending is when you took mom and dad and you just mashed their traits together. What would that look like?
Half and half, kind of?
Yeah. You can think about it as half and half, like half peas, half yellow, half green. Or what else would you think of?
Maybe all yellow or all green.
Well, it's a mixing of the parental traits.
Yeah.
Did you like just kind
Oh,
of
half.
breed the kids together?
I'm not sure what you mean by that.
So like the peas half green, half yellow, like pea, 1 pea.
Said one would they change?
Yeah, we have a brand new color. I don't know what color was green and yellow would give you.
It'd just be a lighter green or blue.
Lighter green. So they would be like a lime green,
Yeah.
like a bright fluorescent green, right? Right. That's what blended would look like, right? Now the other hypothesis, the homuncula process, would show you that that it would just be following the father, right? It would be all yellow peas in the final generation, right? So disproving things. Now, Mendel also performed reciprocal crosses. So he took pollen from yellow peas and eggs from green peas, and then he did the vice versa. So taking males of green peas and females from yellow peas. So just making sure the sex of the individual didn't matter, and ultimately it proved to give him the same 3-to-1 result. So again, that's an important thing, right? As you guys probably know, there are traits that are inherited on sexual XY, right? You guys probably know that color blindness is predominantly found in men. Why? Because it's on sex chromosomes, and the Y chromosome is basically a useless pile of DNA. Not it does stuff when we develop, it just doesn't do anything after. So Mendel, basic monohybrid. What do you learn from that? Well, Mendel learned a couple of different things. First, he proposed that each plant carries 2 copies of a unit of inheritance. Now, remember, DNA wasn't discovered until 1950. Watson and Crick, and Rosalind and Sharp. Right? So 100 years before this discovery. And so what he proposed there was traits. He didn't know what they were. He didn't know what carried these traits, but he knew that they were. Traits have 2 forms that can each breed true. Now, we know those as genes, but he didn't know that. Traits that appears in the F1 progeny is the dominant form, and the traits hidden away in the F1 progeny is what we call the recessive form. Should sound familiar. The units of inheritance are known as genes. He didn't quite know that, but we know that now. And alternate forms of a single gene should be recognized as an allele. Right? And alleles can be subtle. They can be 1 base per change, or they can be a massive gene. But it's some variation of the gene. In individuals with 2 different alleles for a single trait, we define as monohybrid. And our gametes, as again, you probably remember this, but we're just going to do refresh your memory. They're the specialized cells, sperm, egg. There's weird stuff in fungi, but think about it as like sperm and egg that carry the genes across generations. Right? It's not like bacteria where I cut my skin cell off and it grows into a new person, right? Sperm and egg have to come together. Right? So exciting. Nothing, like I said, this isn't it's not new information, but it's it's foundational for what I'm talking about for the rest. Now, the important thing, all the things we talked about are important thus far, but Mendel came up with the law of segregation. And the question he asked about this is, how do progeny inherit 1 unit from the maternal parent and the other unit from the parental parent? How do you for mom. You have two ideas. Well, two things. The two alleles for each trait separate during gamete formation. So we have a big Y, so dominant yellow P, and a little y, recessive P. They're both pure breeding. They grow into a plant and they produce gametes. Now, these pure breeding plants only have one type of allele, big Y or little y. So the gametes that they can produce, well, they have to be only carrying the big Y or the little y, right? They're pure breeding. Now, if we look at the fertilization event where we fertilize big Y, male or female, doesn't matter, with little y, we produce a hybrid, right? A big Y, little y, a heterozygote. That seed develops into a plant, producing us an F1 hybrid that has a yellow P showing the dominant trait, but it still has that recessive trait as genetics. So nothing crazy, right?
Good question. When he was crossing them over, did he know what the sperm was and what the male was?
Well, he could tell
The
the
female and male.
and so he could tell the trait, right? Now, sperm is pollen in terms of these plants, right? So that's relatively easy to distinguish. And you remember back from a few slides ago, you can look at the anatomy, right? The ovules are hidden deep in the stigma itself. So
Okay.
you don't really, it's all, the way it works, right, is you collect pollen and then transfer. And then
Okay.
once you're done transferring, you snip off the anther, so nothing else.
Okay, cool.
And so we can conceptualize Mendel's ideas with the Punnett square. And you guys probably, again, you probably know this. You might hate it, you might love it. I personally like Punnett squares quite a bit. They're fun to do, especially the big ones. But a Punnett square is a very simple way to visualize Gregor Mendel's results. So each F1 hybrid produces two kinds of gametes in a 1-to-1 ratio. So if you think about this scenario, the parental generation was big Y times little y. Offspring, they produce, the gametes they can produce are only one type, either big Y or little y. Again, they're pure breeding. Your F1 generation is all identical because they're a mixture of mom and dad, big Y, little y. So they're heterozygous. Heterozygotes can produce two distinct types of sperm and two distinct types of eggs. Because they're heterozygote, they can either pass down big Y or little y in their sexual cells. So when we start to draw a Punnett square, we think, okay, the ratio of gametes that get big Y, the ratio of gametes that get little y, is about a 1-to-1 ratio, or about 50% of the total gametes. So when we're drawing a Punnett square, we essentially stack here and here. Again, this is just me refreshing everyone's memory. With one potential gamete from mom and one of the potential gametes for dad. And the same thing for the eggs as well. And we use the Punnett square to basically fill in, right? So if I have a big Y here, I donate that, and then it's fertilized. It fertilizes this egg, which is also big Y. So I have an individual that's big Y, big Y. If I donate this allele, it's little y, and I get this allele, it's big Y. I have little y, big Y, the heterozygote. And the same thing here. And then in the final box, little y donates, little y donates. We get a little y guy, which is how we get that 3-to-1 ratio that Mendel. So remember, in this case of classic Mendelian genetics, some traits are dominant to others. Typically speaking, we express the dominant trait as a capital letter. In this case, the dominant trait is yellow, recessive trait is green. Yellow gets donated by big Y, green gets donated by little y. Now remember, the dominant trait masks what we see in the recessive trait. So if one dominant allele is present, it completely masks the recessive allele. It's only when you have two recessive alleles that the recessive allele is expressed. Now remember, the way this works is, even though you have two copies of every allele inside your cell, you basically inactivate one chromosome. You only express one of those alleles. That's typically how most traits work, which is why you have to have two recessive for you to express it. So now Mendel's Punnett squares reflect the basic rules of probability, so fun statistics. Did I go over time already? Yeah. Is this class ending 11?
11:40.
Yeah, no, it stops at 10:45, starts again at 10:50, and then goes to 11:40.
All right. I
Technically.
got more time. I thought it was. She said I thought I
So
ended at
11:30.
Yeah.
All right. So the product rule, which you might remember from math classes you've taken here previously, or maybe from high school, wherever. The probability of two independent events occurring together is the product of their individual probabilities. So we ask the question: what is the probability that event 1 and event 2 will occur? So that's the product rule, which we represent as P as in probability, not parental, but probability of 1 and 2. So these are two independent events. They're not relying on one another to occur. It equals to the probability of event 1 times the probability of event 2. So if it's 1/2, it's 0.5 times 0.5. It's the product rule. And I'll be perfectly honest with you, I confuse these all the time in my brain, so don't feel bad if you it's in your brain. But we have the next step, which is called the sum rule. Occurring is the sum of their two individual properties. So the question we ask there is, what is the probability that event 1 or event 2? Now it's an important distinction. And together, 1 or the other. That's the difference here. So the probability of 1 or 2 is the probability of event 1 plus the probability of event 2. Okay, this is the most complex of statistics we'll get. But like I said, I've been doing this stuff for years, and I always mix these up in my head, so I do not blame you if you mix them up in this one. So let's apply this to Mendel's rule. So we have a scenario. We have a hybrid cross, big Y, little y times big Y, little y p's. So what is the chance of getting offspring Y? Well, the chance of Y pollen, big Y pollen, I should say, is 1/2. Well, why is it 1/2? Well, this individual has a 50% chance of donating the big Y, and this individual also has a chance of donating a big Y, i.e. producing an embryo or a sperm with big Y as the allele. So the chance of both of those, the pollen is 1/2, the ovule is 1/2. So the chance of Y pollen and Y ovule is 1/2 times 1/2, or 1/4. Now, back to the Punnett square, what do we see? Big Y, 1/4, right? 1/4 of these potential offspring. That works. So what is the chance of getting a Y, big Y, little y, so a heterozygote? Well, we know the chance of pollen uniting with a little y ovule is 1/2 times 1/2, giving us 1/4. And the chance of Y pollen uniting with the little y pollen uniting with the big Y ovule is 1/2 times 1/2, or 1/4. Now, the sum rule tells us the chance of either event happening is 1/4 plus 1/4, giving us a 50% chance. Now let's visually look at that. What do we get? 50% chance. 2 of every 4 new individuals produced has a chance of being, or 50% has a chance of being a heterozygote. So I find that a lot of students don't like this stuff. They prefer to draw it up Punnett square. And I recognize that. Punnett squares take time. And so you should try to become familiar with this idea as best as you can. Just because it'll, I don't know. I mean, sometimes it's conceptually better to look at a Punnett square. But some people, it's conceptually better to look at statistics or probabilities. So it just depends what you prefer. But definitely try to get comfortable with this as best you can. So let's look at more crosses from Mendel to further differentiate this law of segregation. So Mendel took his F2 plants, were then self, i.e. he crossed a plant with itself to produce the third filial progeny, or the F3 generation. Now, when Mendel looked at his ratios, all of the F2Ps were pure breeding, right? Because it's, you're doing yourself 1/3 of the yellow F2Ps were pure breeding. Sorry, the green peas were pure breeding. 1/3 of the yellow peas, F2Ps, were pure breeding. And then 2/3 of the yellow F2Ps were hybrid. So let's break that mess of sentences down. F1, heterozygote. It produces 4 potential offspring in that 3-to-1 ratio. Remember, that 3-to-1 ratio is the phenotypic ratio, where we have 3 yellow to 1 green. Now, from a genotypic standpoint, it's 1 to 2 to 1. That's the ratio. Now, when we look at what happens when you self-cross these, so big Y with big Y, little y with little y, and so on and so forth, what you produce is, when you cross big Y with big Y, you get all big Ys out of third generation. Makes sense, right? We drew out a Punnett square for that. That's the only thing that would make sense here. Now, when we do another hybrid cross, big Y with little y times big Y with little y, our third generation is again that 3-to-1 ratio. 3 greens, so 3 yellows to 1 green. Same thing when we took this other offspring, big Y, little y. We get the same exact 3-to-1 ratio. Again, that's a phenotypic ratio, not a genotypic, but a phenotypic. And then when we cross the green plants, we only get green plants out the back. So that's where we're at. So makes sense through multiple generations. So let's get into some definitions. I've thrown these out and I've defined them loosely, but let's formally define them right here. Phenotype is any observable characteristic. My receding hairline, oh so sweet phenotype from my ancestors, those Irish jerks. The genotype is the underlying genetics that give me my receding hairline. Yeah, those jerks. A homozygote has 2 identical alleles, and they can be homozygous dominant, i.e. having 2 big alleles, big Y, or homozygous recessive, having little y, little y. And then finally, our heterozygote has 2 different alleles. For example, in this case, it's big Y with little y. So it has 2 distinctly different alleles. And in this case, we're thinking about the basics, so there's really only 2 alleles to think about. We're going to get more complex where we talk about 3 alleles or more. But for now, we're just thinking about 2 distinct alleles.
I have a quick question. It does not have to pertain to this. If you're at Mass General, what phone number? It'll pop up as it won't pop up as Boylston, right? Or will it? I'm supposed to pick my aunt up in the house. She's supposed to call me when she's ready. I thought it was gonna happen until like 3 o'clock.
To be honest, I have no idea.
Do you mind if I run home?
Yeah, it would.
Yeah, right?
Yeah.
Because it says Boylston.
Yeah.
And so remember, the heterozygous phenotype defines that the dominant allele, in this case, our yellow is the dominant trait we're thinking about. So the dominant allele, big Y, is dominant to the recessive green allele, little y. And a dominant allele with a dash represents an unknown genotype. So for example, we have Y dash that stands for either big Y, or big Y, little y. That's all. Now, when we start to look at the genotypic differences, the genotype versus phenotype in those homozygotes and those heterozygotes, we start to see some interesting things. So we have a cross of 2 heterozygotes, big Y times little y. The genotypes in the F2 generation, remember, they're in that 1 to 2-to-1 ratio, genotypical. 1/4 are big Y, half are big Y, little y, and 1/4 are little y. Remember, the phenotypic ratio was on the F1 and the F2 generation was 3-to-1, right? But the genotypic ratio is slightly off. And that's because that dominant allele suppresses recessive. And the phenotypes in the F2 generation are all 3-to-1. 3/4 yellow, 1/4. The F2 generation are 3/4 yellow and 1/4. So you can have a disconnect between the genotype and the phenotype because of heterozygote. So we could use a test cross to reveal an unknown genotype. So we can ask the question, is the genotype of an individual with a dominant phenotype, for example, big Y something, heterozygous or homozygous? Well, the solution here would be test cross a homozygous recessive to examine the progeny. So our cross would look something like this. If we don't know if we have heterozygote, so big Y, little y, or homozygous dominant, big Y, we could do two distinct crosses to figure this out. We cross big Y with little y, 100% of the offspring should be heterozygous. So we should only see the yellow trait present. Now, cross B, if we had a heterozygote, so big Y, and we cross that with a homozygous recessive here, so little y, that resulting cross would produce 50% of the offspring would be yellow and 50% of the offspring would be green. So we can use this, again, this test cross with the homozygous recessive to figure out a potentially unknown genotype of a dominant individual. So pretend if I gave you, you have a yellow pea plant, it's big Y something, and I want to know if it's homozygous or heterozygous. Well, you just do a simple test cross with the recessive plant to figure that out, and the resulting progeny will tell you that. If it's dominant, well, you'll see only yellow plants. If it's recessive, you'll see a mixture of yellow. So, Mendel. We're going to keep talking about Mendel because why would we stop? We've only been talking about Mendel for an hour. So Mendel's crosses revealed the law of independent assortment. So I've thrown that idea around. I've shown you the evidence for it, but what does it mean? What is it? Right? So Mendel tested the simultaneous inheritance of multiple genes using what we call dihybrids, i.e. plants that are heterozygous for two distinct genes. So it was had green plants, green peas, and they were wrinkly. Yellow peas and they're wrinkly, right? Two distinct traits. That's what I mean by dihybrid. So first he would cross true breeding yellow round peas with true breeding green wrinkled peas. Now you'll see that yellow and round, yellow defining as big Y, sorry, just big Y, and round being defined as the dominant trait with the letter R. We cross those with yellow, with green peas, which we know is the recessive. But wrinkled is also recessive, which we define as little r. And so that cross of two homozygous dominant, so two pure breeding lines, dominant versus dominant times dominant, recessive, gives us an F1 generation that is a mixture of both genes, right? We have a hybrid, right? A dihybrid. Big Y, big R, little r. So if we do what Mendel does, and we take those F1 plants and we self them, so we have two heterozygotes. We have a heterozygote from F1 crossing with another heterozygote from the F1 generation. The F2 generation is produced.
So I don't know if this question is going to sound very stupid, but I know you said that, like say with the color blindness, that it's like heads on the X or Y. So when it comes to the dominant and recessive, is it possible to have like a big Y, little y for like the yellow or whatever, but it's like only on one sex? That it does the recessive like show or something like that?
So there's a couple things about sex inheritance. So remember, the Y chromosome lacks meaningful genetic information. So if you have an allele on the X chromosome, it's not backed up by another allele on the Y chromosome. So it only takes one allele to make a trait if it's sexually inherited. And we're thinking about humans and flies and normal breeding systems here. Now, in terms of how X chromosomes work, you should recall that X chromosomes are inactivated. So you only have one chromosome working. So we know that X chromosomes are inactivated by mRNA or microRNAs. Like they wrap around the X chromosome, inactivated. So when you're thinking about X, sexually inherited, you really only think about one chromosome functioning at the same time. Now, for males, they only have one functioning chromosome in general. Right? So if something's present on the X chromosome for a male, an XY male, you could be an XX male, yeah. YYY. There's all sorts of weird males. But only with a functioning X chromosome. And the Y chromosome is just really for forming testicles. Gotcha. So this is the cross we have. Two heterozygous for two distinct traits crossing with one another. And Mendel asks whether those F2 progeny would be parental types, yellow round and green wrinkled, or would some be what we call recombinant types, yellow wrinkled and green round. But right now, all we're seeing is in the parental generation, yellow round, green wrinkled, and the offspring generation, we have yellow round. We haven't seen that mixture yet. And Spoiler, the answer is yes. You should recognize this cross. So this is a figure from a textbook, but it's a really great figure. We have this dihybrid cross. Cross produces parental types and recombinant types. So we have of 16 total plants produced. You can extrapolate that to be any number you so choose. You will produce, in the case it's 315, 108, but we'll just talk about the ratios. Out of 16 plants totally produced, 9 will be yellow and round. 3 will be green and round. So again, recessive mixed with the dominant. 3 out of 16 will dominant trait, and then 116 will be green and wrinkled, 2 recessive traits. So we get the beautiful 9:3 to 3:1 ratio when we have a dihybrid cross. So we can look at the ratio of yellow to green. It's 12:4 or 3:1. That's what we observed in our single crosses, right, from a few slides ago. And we can look at the ratio of round, which is the dominant trait, to wrinkled, recessive. It's also 12:4, which is a 3:1 trait, right? Just like we saw previously. We looked at a single hybrid cross, right? We looked at big Y, little y times big Y, little y. The ratios are there, but when we start to mix them, we see a 9:3 to 3:1 ratio. Now, this is phenotypically speaking. There are differences in genotype here, right? And so Mendel's independent assortment in the crosses of these F1 dihybrids produces that 9:3 to 3:1, right? 3:1 ratio. Try saying that 10 times fast. And so each F1 hybrid produces 4 possible gametes in a 1:1:1 ratio. And how does that work out? Well, if I have, this is my parental generation, big Y, big R, little r, big Y, little y, little y, little r, little r. Okay, don't try to say that 10 times fast. The gametes that they produce can only be big R, or big Y, little y. Right? That's how the gametes work out. Now, their offspring, which are dihybrids, have the potential to produce 4 unique gametes. They can produce, because again, we're starting off with this, right? Heterozygote. Big R, big Y, little y. Potentially, you can donate to this system a big R and a big Y, a big Y and a little R, a little y and a big R, or a little y and a little r. You have the potential to produce 4 distinct types of gametes, all in 1:1:1 ratio.
So because that they're the same on both sides, can't you just kind of like reduce it a little bit to like just the 1 big R, little r? Or do you have to keep the little y, big R?
Because remember, we're thinking about 2 distinct traits here.
Oh, okay. Yeah.
Right? We have to have, because you can have, remember, because if you're trying to mix this up together, right? Well, if I donate the big Y, I can either donate this or this.
Yep.
Right? If I donate the little y, I either get this or this, right? And vice versa. So there's you have to basically look at all potential combinations, which means there's 4 total. And so we get this significantly larger, 4 times larger Punnett square from our dihybrid cross. And again, this is how we get our 9:3 to 3:1 ratio. You can visualize it, right? 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3. 1, 2, 3, and 1. Right? That's how you visualize that. But you notice that the genotypic ratios don't match that, right? We only have 1 big Y, 1 big R, right? And 1 little y, 1 little r, little y, right? And there's a whole big mixture of other things. Lots and lots of different types of heterozygotes. But again, this is exactly like our simple Punnett square, just scaled up these slightly larger. Now, let's put the math behind this. During the formation of gametes, different alleles segregate independently of one another. So big Y is just as likely to assort with big R as it is with little r. So they're not dependent on one another. So if I'm going to get big R, I have an equally likely chance to get big Y or little y with it. It doesn't matter. It sorts independently. That's important. And the same thing can be said true with little y. Little y is just as likely to sort with big R as it is to sort with little r. That's an important piece to remember. Your genes sort independently. They're not inherited together. Now, we'll learn later in the semester that the closer two genes are onto a chromosome, the more likely they are to associate with one another. But for the purposes of what we're talking about here at the basics, genes sort independently in the production of gametes. So we can use the product rule here to calculate the ratios observed in the F2 generation of a dihybrid cross. So each trait is inherited in a 3-to-1 ratio. So the ratio of yellow to green is 3-to-1. The ratio of round to wrinkled is 3-to-1 as well. And that's what we see here. Now, the probability of something being yellow in wrinkled is the probability of yellow times the probability of wrinkled, which is 3/4 times 1/4, giving us 3/16. And if you look at our Punnett square, it's 3/16. I know, not exactly this. Punnett squares are significantly more safe, right? But at the same time, it's good to show the math. Think about what would be faster to calculate this product or to draw an entire Punnett square. Math would be faster. You just have to think about the math. But again, you do whichever you feel is most comfortable to you as an individual. And we can also do and you'll do this in your lab as well for your crosses that you're going to do. But you're going to do what we call a branched line diagram. The progeny phenotypes for each gene are shown in different columns, and this gives the same ratios as seen in the Punnett square. We have gene 1, and we have gene 2, and we have the phenotypes. So we know gene 1 is 3/4 yellow and 1/4 green. Gene 2, again, it's 3/4 round, 1/4 wrinkled, 3/4 round, 1/4 wrinkled. That 3-to-1 ratio that we've been discussing. The phenotypes, 9/16 have to be yellow and round, 3/16 have to be yellow and wrinkled, 3/16 have to be green and round, and 1/16 has to be green and wrinkled. Right? Now, so everybody feeling good with Mendel? You're going to get a lot of practice with this in the lab next week, so don't feel like you have to know all of it. You're going to do some of the stuff in your problem set this week as well, but don't feel like you have to know everything what I'm saying. The terminology should feel relatively comfortable. And very much like we did a test cross on a hybrid cross, we can also do a test hybrid on a dihybrid cross. And so if we don't know a genotype of an individual, right, if we have big Y dash, big R dash, we can do a test cross very much the way we did a test cross. We simply just need to do 4 test crosses instead of just doing 2. And this shows you how to go about doing that. I'm not going to walk through it. We already walked through 1 test cross. Take a peek at this if you want to go into how to do it. But again, nothing crazy. And so now we start getting into some slightly more complex things about Mendel, but not too complex. Mendel, you can use his crosses and his laws to predict the offspring from complicated crosses. So the number of different gametes equals 2 to the n power, which n is equal to the number of different traits. For those of you that have done PCR before, that 2 to the n should sound familiar. And we'll cover that later in the semester in lab. But 2 to the n is a pretty common mathematical thing in science. So if we have the number of different gametes is 2 to the n, with n being the number of different traits. So we have 4 distinct traits here. 2 to the 4 gives us 16 distinct kinds of gametes. Conversely, if we have 1, 2, 3, 4, 1, 2, 3, 4, 16 times 16, because we have 2 to the 4 times 2 to the 4, 16 times 16 gives us a distinct combination of 256 genotypes. So this is like, I have, 4 genes involved. Now, we're mostly talking about similar, simpler crosses, but some traits are controlled by dozens of genes. So we know that, for instance, really complex disorders like autism are controlled by potentially hundreds of genes. So you can imagine these numbers get big, pretty big. But to put this in perspective, if you're going to do a Punnett square of this, so this cross here, you would need 16 columns and 16 rows. It's a monster Punnett square. I need the entire board. So the easier way is just to break this down into a multi-hybrid cross into independently assorted monohybrid crosses. Just as it. So let's just break that down. So we can predict the proportions of progeny from multi-hybrid crosses. This is just one example. So the crosses, these hybrids times this hybrid, what proportion will be AA, BB, CC, and DD, right? This funky mixture. Well, we break it down. We know that little a, big A times big A, little a gives us 1/4. We did a Punnett square of that, which shows 1/4 would be big A. We satisfy that. We do the heterozygote times the heterozygote for B, for C, and D, and we can get their associated probabilities. Now, we use our product rule because we want to know what is the probability of this, and this, and this, and this, not or, but it's and, right? So we have to multiply the probabilities. So it ends up being all these probabilities, 1/4 times 1/4 times a half, which gives you 1/64 of all your potential progeny are this specific genotype. So again, I know math sucks. What's faster, doing this or doing a 16 times 16 Punnett square? I don't know about you guys, but even sometimes when I used to do when I took genetics 10 years ago now, I used to sometimes make mistakes on the 16 box Punnett squares, like the hybrids, let alone try to do a Punnett square of this monstrosity. Just as a note, you can apply really simple mathematical formulas to figure out what is the probability of your next generation. We can do the same thing here. This multi-hybrid cross, how many progeny will show the traits for big A, big C, and big D, and have the recessive for B? Well, we're looking at 4 distinct monohybrid crosses here. That's all these multi-hybrid crosses are. We know that when we cross these heterozygotes with one another, the probability of having the dominant A is 3/4. The probability of the dominant C and the D also has to be 3/4. And the probability of getting little b is 1/4. Right? Because you can just do a Punnett square out for those things. So the expected proportion of A dash, little b, C dash, and D dash is 3/4 times a half, 1/4 times 3/4 times 3/4 gives us 27/256. So again, huge Punnett square or a little bit of math. Conversely, you just do 4 small Punnett squares and you get the probabilities. Or you can just visually look and say, hey, this is a hybrid cross, so it has to be 3-to-1. Again, I don't want to tell you how to do things, but whatever is most comfortable for you as an individual. I know some people just really prefer the visuality of Punnett squares. That's okay. But a Punnett square of this magnitude is clearly not possible. So Mendel did all this stuff in the 1850s. Now, as we discussed, DNA technology didn't really take off till Watson, Crick, Chargaff, and Franklin elucidated the structure, right, of DNA. And I keep saying those 4 names because Watson and Crick always get the credit, but Franklin, badass woman in science, Chargaff, awesome dude in science, so they deserve credit equally as much as Watson and Crick. So Mendel's work was published in 1866, but it wasn't until almost 40 years later for scientists actually to independently rediscover Mendel's work. And it wasn't until they discovered Mendel's work and these scientists were familiar with Darwin's work that they put them together. I started off, when I started talking about Mendel, I mentioned how similar this is to Darwin, because again, they're 2 sides of the same exact coin. It wasn't until some people Karl Korens, Hugo de Vries, and Erich von Schmark. I don't know. He's German. German names are hard to pronounce. Wasn't until these so handsome men with, varying levels of good beards, no sweet mustaches. Mendel being the odd guy without the mustache or the beard. I don't know. I feel like all pictures from this time, everyone had a mustache. And so it wasn't until they took Mendel's ideas and said, "Holy crap, this works. This was discovered 40 years ago." And then they put them together with Darwin's ideas, and then it started to make a lot more sense. And so the important thing is, okay, we can look at probabilities, look at all this stuff, but what is the underlying mechanism here? It's DNA. And what does DNA encode for?
What was the question?
What does DNA encode for? Well, yeah, it codes for you, but more specifically. DNA encodes for genes. The DNA encodes for messenger RNA, which has potential to make protein. Right? So the central.
I feel like I thought that was a lot more complicated.
No. As a general note, when I ask questions, they're usually pretty simple. One of the things I've learned about students is that students always think what I'm asking, what any teacher is asking, is far more complicated, but it's not. Yeah, so we're thinking about that central dogma of biology: DNA, RNA, protein, right? Mendel didn't know that. We didn't know that, but at the time, in modern times, we know that, right? So let's think about what Mendel's experiment looks like, but from a molecular point of view. So I think that's important. You think about traits all you want, you think about genes, but there's an underlying chemistry, biochemistry, protein, right? Here. So the big R allele encodes for starch branching enzyme, SBE1. The little r allele does not make SBE1. So what ends up happening is sucrose and unbranched starch build up, leading to wrinkled peas. Remember, this is encoding for shape, not color here. So the biochemical function of Mendel's pea shape is big R is a functioning SBE1, little r is a non-functional SBE1, or a none, which leads to a couple different things. So we have the buildup of amylose, just as you can tell, it's just a sugar, just a byproduct of photosynthesis and stuff like that. Those that have the dominant allele, either big R or big R little r, produce from that amylopectin, giving them that nice round shape. Those that have little r lack the enzymatic machinery, again, SBE1, to convert amylose into amylopectin, leading to a buildup of amylose or starch, which gives them their distinct wrinkled pea shape. It's pretty simple, right? One enzyme controlling, or one gene controlling one trait because it produces one protein, one enzyme. With that enzyme, we have a chemical reaction converting amylose into amylopectin, giving that distinct. Really nice. There's lots of examples of this in nature. We're going to look at sickle cell anemia, or sickle cell disease, I should say, in lab later this semester. A nice, simple, one gene, one protein, one trait. Now, let's look at the big Y, so the color. Big Y encodes for the stay green enzyme. Not exactly the most exciting names here, but they're descriptive. SGR. I always, in my brain, I call it Sarge, but Sarges would be SRG. And this helps break down chlorophyll. You guys should know chlorophyll is the pigment of photosynthesis. Now, the little Y allele does not make SGR, so chlorophyll is not broken down, and the peas remain green. Peas, you can imagine peas, which are seeds, they don't need to photosynthesize. So having that chlorophyll is kind of a waste of space, right? So we have chlorophyll, looks a little bit like this. In the presence of a functioning SGR gene, so big Y or big Y, little y, it cleaves chlorophyll into this. You can see it just breaks the ring, which causes chlorophyll to lose its magnesium core, loses its color, leading to the presence of yellow peas. Now, little y, it has a defective or non-functioning SGR gene, which means the product is not broken down, which leaves chlorophyll nice and happy and circular and green. Thus, the peas are. Phenotype has an underlying biochemistry, which has an underlying genotype. That's an important thing. Phenotype has a genotype and an underlying biochemistry. So, two general molecular principles that we want to think about. A specific gene determines a specific protein whose activity may affect the phenotype. And two, a dominant allele usually determines a normally functioning protein, and a recessive allele usually does not encode for a functional protein. Those are generalities. Now, we know there are many genes out there that encode for just messenger RNA, and that messenger RNA does something. It doesn't make protein, it just does something. So there are genes that out there that don't necessarily make a protein to do their action. So there are caveats to number one. And we also know there are caveats to number two, where the dominant gene is not necessarily the best gene. So the disorder you're probably familiar with is Huntington's disease. The dominant gene makes a defective protein, which causes a really bad disease. Whereas the recessive gene causes a nice, happy functioning gene, which does not lead to a terrible. So these are the general rules, but not always the case. And in terms of how Mendel's stuff applies to humans, it works pretty good for some of our traits. You know, Mendel's rules don't just apply to peas. You can look at Mendel's rules and apply to anything that undergoes sexual reproduction. So what we're talking about here does not apply to like bacteria or fission yeast, but it applies to anything that produces some form of sperm, some form of egg. That can be anything in wacky and wild in the animal world or the fungal world or the protost world, but Mendel's rules don't just apply to peas. So many inheritable traits in humans are caused by the interaction of multiple genes, and so they don't show typical Mendelian inheritance patterns. As of 2016, there were approximately 6,000 single gene traits known in humans. That number has gone up slightly, but not hugely since then. And even with single gene traits, determining the inheritance pattern can be very, very tricky in humans relative to peas. Humans are a terrible model organism. The reasons we don't study humans for everything in the world is because we have a really long generation time. We produce a small number of offspring, even those crazy like octomoms, decamoms, and crazy moms. My sister-in-law's boyfriend's mom, if I'm not mistaken, the transition there, had the grandmother had 64 kids, which is just insane.
64, like all she gave birth
Did
to?
she push like one?
Were they
No,
all
not at
twins
once,
and
but like
quadruplets?
she had like several quadruplets, several triplets, several lots of single kids,
Gotcha.
lots of. It was crazy.
Both my grandparents on my mom's side had 6 kids. It was twins and the triplets and then one, and then it was like triplets, one.
Can you imagine that?
Damn.
Yeah.
64.
After the first one, I will have my things snipped.
Yeah. I'm not even giving birth. Like, I have no, obviously, I have no potential to ever give birth, but I'd be one and done. I would be one and done. I don't know about you ladies, but I would be one and done. Ooh. Anyways, so humans are terrible. So understanding Mendelian inheritance in humans can be kind of tricky. But here are some things caused by recessive alleles. Thalassemia, it's on chromosome 16 or 11. It's reduced amounts of hemoglobin. Sickle cell anemia, which we'll do a lab about, which has abnormal hemoglobin. It's on chromosome 11. Cystic fibrosis, which is on chromosome 7, which has defective cell membrane proteins. Tay-Sachs disease, chromosome 15, and phenylketonuria or PKU, all single gene inherited things with different levels of probability being found in the population. Single genes, still kind of hard to elucidate things. Now, in addition, there are some dominant single gene traits, including hypocholesterolemia, chromosome 19, Huntington's disease on chromosome 4, differing levels of probability. And also, it's kind of interesting, it's different levels of probability depending on the population you are found in. So if you're, a French Canadian, the probability of having hypercholesterolemia is much higher than it says if you have ancestry from, say, Africa. The same thing can be said for other things, like for instance, our friends with sickle cell anemia, probability in African Americans is significantly higher than it is in, say, people with Europeans.
People with Huntington's disease, I know it's like Caucasian. Is that common with people that have had like things like radiation? Like,
So Huntington's is probably the scariest disease in my opinion. So Huntington's arises either from a defective chromosome 4, so you have the dominant allele, or there's spontaneous Huntington's, where you have an in vitro or a in situ, so just an irregular body mutation. But most commonly is genetically inherited.
Oh.
So it can be random, but it's most commonly inherited. And so, because human genetics is a pain in the butt, we use pedigrees, and again, probably something similar to what you've heard or seen before. The orderly diagrams of a family's relevant genetic features. It includes as many genetic generations as possible, ideally at least both sets of grandparents of an affected individual. And you can analyze them using Mendel's laws. It can determine whether a trait is by alternating alleles or of a single gene, or is it dominant or recessive. So typical common symbols we see for human pedigrees: squares are males, females are circle, and these are biological speaking. And then sex as unspecified as diamond, so like you just don't know. Doesn't mean like transgender, it just means you just don't know. Like it's, oh, this person existed, but we don't know their gender. Typically filled in circles are diseased. Open circles typically are undiseased. You can see deceased has a slash through it. Shoot, I can't remember what this means off the top of my head. I'm so sorry. Anyways, the double line means consanguineous, meaning I can't say it. Multiple progeny is denoted by the number, so 14. And then you can look at generation 1. There's like a branch. We have the subship line, so we start to see individuals that are brother and sister, or brother or sister, so on and so forth. So this is the basic anatomy. I'll have to look this one up. I'm so sorry.
It's not like married, like would it be like a married or mating couple with parents?
I'm sorry. I don't want to give you the wrong information. Sorry. Anyways, it's not that important. Just to understand like the anatomy of it. Let's look at this pedigree. So again, a pedigree is simply a vertical pattern of inheritance indicating a rare dominant trait. In this case, we're looking at Huntington's disease. So we can see that every affected person has at least one affected parent. Mating between affected persons and unaffected persons is effectively a test cross. Nice. Yeah, so it's
It would be like
yeah,
somewhat
like,
relative.
like cousins.
Yeah, cousins. Okay. The type of thing.
So you can see we have female has it, male doesn't. They produce they produce 2 offspring, both of which have it, and then they come together and so on. So you can track the disease through time, and just by looking at how it's inherited, you can start to piece apart. Well, how does this work? Right? What would be the underlying Mendelian genetics of this system? Also, this poor person had 14 kids, right? But clearly, this is present way too much throughout the tree to be the pedigree to be physically a recessive trait, right? There's just too many kids in this 4th generation that have the disease. Right? You have 6 offspring, and 4 of the 6 have it. So clearly, this has to be a dominant trait. But this is what a pedigree looks like. Now, Huntington's, as I've said, is probably the scariest genetic disease out there. Huntington's, the dominant Huntington allele produces an abnormal HTT protein, and this abnormal protein damages nerve cells even when the normal protein is also present. So you can be heterozygous dominant, like we have here. I'm sorry, the normal HTT protein here, or the defective HTT protein. And you have to have 2 copies of the normal allele, which are going to be recessive. But if you have even 1 copy of the dominant allele, it's going to override the functioning of the recessive allele even being produced, which is why Huntington's is a jerk. Those of you who don't know, Huntington's is late onset, so it usually onsets when you're in your 30s and you die before you're 40. Why Huntington's is such a terrible disease is neurological, and you can pass it on to your kid without even knowing you had it. It's really kind of bad. It's more common in like Northern European folk. So, and you would know someone in your family had it. So it's not like it pops out of nowhere. Most of the time. And so we can look at another pedigree. So we have a horizontal pattern of inheritance, a rare recessive trait. In this case, it's cystic fibrosis. So you can see we have 2 parents, they have some offspring, the offspring have offspring, and we go all the way down to 7 generations. Well, you can see that the cystic fibrosis being recessive doesn't pop up at all for the first 5 generations here, but it does pop up in the 6th and 7th generation. Cystic fibrosis is another disease. You know you have it. It's not like it's like hanging around. You have cystic fibrosis, you have cystic fibrosis. But you can see that if you use the pedigree to track, and just given how rare this trait is in the pedigree, it just simply has to be a recessive trait. It just has to. Cystic fibrosis is caused by a normal CFTR protein, cystic fibrosis transmembrane receptor, if I'm not mistaken. And this regulates the passages of chloride ions, so just Cl minus in and out of the cellular membrane. Heterozygous individuals produce enough CFTR for normal function, whereas homozygous recessive simply do not, which means they have a buildup of chlorine ions inside of their cell, which leads to a buildup of mucus outside of their cells, principally in your lungs, which makes those that have cystic fibrosis very, very difficult for them to breathe. They generally don't live past like 25 or so, which is the... So how do we recognize dominant traits in pedigrees? 3 key aspects. Affective children always have at least one affective parent. As a result, dominant traits show a vertical pattern of inheritance, and 2 affective parents can produce unaffected children if both parents are heterozygous. How do we recognize a recessive trait in pedigrees? 4 key aspects here. Affective individuals can be children of 2 unaffected individuals, particularly as a result of between relatives mating. That's what that word means. All of the children of 2 affected parents should be affected. Rare recessive traits show a horizontal pattern of inheritance, and recessive traits typically show a vertical pattern of inheritance. What do I mean by horizontal and vertical? Horizontal means like when you bring in new people from outside, and you don't have this within family meeting going on here. That's what I mean by horizontal. So we're pulling in from a distinct family as well. And that's it. So that is the basics of Mendel. That's the basics how to recognize a pedigree. That's the basics of inheritance. Now we're going to go into more depth next week, including doing an entire lab where we're doing some virtual simulations of Mendelian crosses, not dihybrid crosses, just single gene crosses using a fun little computer program. So I will see you guys next week. Please